Story time. There were two individuals, Erisichthon and Demeter. Erisichthon chopped down Demeter's precious tree, which made Demeter furious, and as punishment, Demeter inflicted him with further hunger, which drove him to eventually eat himself.
Now, although self-eating seems like a concept of the past, it is actually a large part of how our bodies reserve the ability to maintain a strong structure. In 1955, Christian Dudov, a Belgian biochemist, identified the lysosome. The lysosome is a cellular organelle that is also known as a janitor of the cell. His lab study of enzymology focused on acid phosphatases, which are enzymes used for digestion. These led him to noticing membrane-bound organelles filled with hydrolyzing enzymes that use water to break down large macromolecules. The impressive part is that this discovery was made by using simply a centrifuge, and yet this centrifuge took him on a path towards the Nobel Prize of Medicine in 1974. Soon after Dudov's discovery, a horde of scientists, including Alex Novikov and Zanvil Cohn, made collaborative efforts to fully identify the more specific functionality of each organelle. But this time, they opted to use visual observation under a light microscope. These experiments concluded that lysosomes act as a janitor of the cell, taking up and consuming damaged macromolecules or organelles in the cell by endocytosis. This led to the idea that cells use lysosomes to quite simply eat themselves, and thus it was termed autophagy after the Greek term for self-eating.
Now that we've learned about how the autophagic process was discovered, let's take a look at some of the present-day definitions and functions of this phenomenon. In the context of the human body, autophagy is the body's way of cleaning out old or damaged cell parts in order to regenerate healthier cells. These healthier cells help the body to work efficiently due to their ability to react and adapt to stress. Autophagy is a main contributor in homeostasis.
I'm a big fan of homeostasis. Do you know what that is?
Of course not.
This process is what helps to maintain steady internal conditions in the cell. More general functions of autophagy include getting rid of harmful invaders such as bacteria, as well as degrading amino acids during starvation for energy. And finally, autophagy can suppress the formation of cells while at other times promoting it. These are important functions which are carried out by the different autophagic pathways.
Three types of autophagic pathways. Microautophagy is the main autophagic pathway, and it's important in removing damaged organelles and unused proteins. When a cell receives a stress signal, an autophagosome, which is a double membrane-bound vesicle, acts as an intermediate that delivers these damaged organelles and proteins from the cytoplasm to the lysosome. The autophagosome then fuses with the lysosome, which contains hydrolytic enzymes, and the content is degraded. Another pathway is microautophagy, where direct engulfment of cargo from the cytoplasm into the lysosome occurs by invagination of the lysosomal membrane. Microautophagy plays a key role in maintaining organelle size, membrane homeostasis, and cell survival in low nitrogen environments. Finally, chaperone-mediated autophagy uses chaperone proteins that form an HSC70 complex. These proteins act as sanctuaries that provide the optimal condition for protein functioning and folding. In this process, these chaperones act to deliver specific sequences of DNA containing the KFERQ motif across the lysosomal membrane receptor, the LAMP2A receptor. Chaperone-mediated autophagy is different from the other two because it unfolds and translates proteins in a one-by-one manner, and it's very selective with which materials actually cross the lysosomal membrane.
There is a lot of debate about whether autophagic-based dieting, called intermittent fasting, is beneficial or not. Intermittent fasting is an eating pattern where you cycle between periods of fasting and eating. It does not say anything about which foods you eat, but rather when you eat them. The idea has become popular and can prove to be beneficial in the future.
Wait, I have a question. Wouldn't continuous autophagic processes deprive the body of essential nucleic acids that are important for other functions?
Also, what's the most effective way to trigger autophagy?
And what happens if someone fasts for too long? How do we know when to stop fasting?
First of all, during fasting periods, the body prioritizes energy reserves over excess nucleic acids to allow for normal functioning and for maintenance of homeostasis. Autophagy can be initiated in many ways, but in this circumstance, glucagon would be the main trigger. And while much research is still required to answer your questions, what we do know is that fasting for 18 to 20 hours is ideal for the glucagon in the blood to trigger autophagy. And finally, it is important to note that autophagy is not only a process that cleans out the cells, it is also a process by which energy can be generated under fasting circumstances.
Now that we know the types of autophagy, let's discuss the relation between autophagy and cancer. Cancer is the second leading cause of death in the world and is characterized by the development of abnormal cells that divide uncontrollably, causing havoc for the body tissue. Given the high prevalence of cancer, the role of autophagy for inhibiting the effect of cancer is definitely an important one. Autophagy has been found to play a dual role in cancer biology. Autophagy can function to promote tumor cell survival, but can also contribute to cell death, a relation that has been called a double-ended sword.